we are. So anyway, thank you again for the chance to give this presentation. I'm excited to get the feedback from the community. So I'd like to begin by describing an overview of the challenge in human neuroscience that we aim to address with the development of this tool. So as we all know, human EEG and MEG are among the most powerful technologies to study brain activity because we can get fine temporal resolution in awake behaving humans. But there's a downside to these technologies in that what we're recording is the so-called macro scale activity that's generated by large ensembles of neurons, and it's still really difficult to infer how are these signals generated at the cellular and circuit level. And this circuit level understanding is really critical if we want to know why these signals correlate with function, and if we want to target treatments when they're disrupted in neuropathology. Now, obviously, invasive recordings and all the advances we have in genetic and modulation techniques in mice and other animal models is ideal for dissecting circuit level dynamics, but these are still really difficult to implement in the human. And so to bridge this gap, we've been developing computational modeling methods. And there are actually two types of modeling that we need to get from the extracranially measured signal to the underlying cellular and circuit level activity. The first is current source estimation modeling. This is where we take the extracranially measured signals and we estimate the location, the direction, and the time course of these big underlying electrical currents that create our sensor data. From there, we need to connect these electrical currents to the underlying cellular and circuit level generators. And this is the ideal problem for computational neuromodeling, where we can simulate the electrical activity of the neurons. And we can do this with specificity, both at this macroscopic recording level and at this detailed cell and circuit level. And so this is the primary focus of my research program. And we've recently turned this modeling framework into a software that we're calling HumanNeocortical Neurosolver that's designed for the community to be able to begin to develop hypotheses and test hypotheses on the circuit origin of human MEG or EEG data. And so today I'm going to be describing this modeling tool and some of the applications that we've been using in our lab to study brain oscillations. So I'll begin by giving you an overview of some of the background on the development of this new modeling tool. And then I'm going to give an example of applying HNN to studies of the mechanisms and meanings of one of the most common brain rhythms known as a 15 to 29 hertz beta rhythm. And I'm going to be concentrating on studies from my lab where we've been looking at beta rhythms in somatosensory perception. And in the end, I'll give a brief tour of the graphical user interface to our computational neuromodeling framework. And I'll describe some other applications and some ongoing expansions that we're working on. So how do we begin to develop a neural model to study the mechanisms underlying these MEG and EEG signals? Well, any model of neural dynamics is, at the end of the day, a system of differential equations that describes how the activity of neurons changes over time. Now, the level of detail that you build into this model depends on the question of your interest. And one of the biggest challenges in computational neuroscience is deciding, well, what's the right level of model to build? And so I'm just going to briefly describe some of the different levels that you can use and then why we chose the level that we did. So at the top of the pyramid here is models with the least level of complexity. These are things like often called neural mass models or population models, where the activity from an entire area is collapsed into a small number of variables. For those of you who may have done some dynamic causal modeling, this is the type of modeling framework that's embedded in that model platform. We could also start to include some of the details of the individual neurons, the spiking characteristics of the neurons, and this can be done with various levels of detail. You can use integrate and fire neurons, you can use detailed Hodgkin-Huxley neurons, and you can couple them in detailed or reduced form. Now, we know that MEG and EEG primarily reflect activity from the cortex, the outer structure of the brain. And so we could start to build in some of the detail of the anatomy and the physiology of the cortical circuit Or because we're building a model, we can include all of the detailed anatomy and physiology of the laminated structure that we want, and there are lots of efforts right now to try to do this. But again, the challenge is deciding, well, what's the right scale for the question that I'm interested in? And what I'm going to describe to you is we've chosen a scale at this intermediate level for HNN, where we include some of the anatomy and physiology of cortical circuits that we think are essential to creating these big electrical currents that we're recording with MEG and EEG. And so to explain why that's true, I'm going to just step back and describe what it is we're recording and how we're connecting these signals to the circuit level generation. So again, when we apply these inverse solution methods to EEG and MEG signals, things like minimum norm estimates, what we're actually doing is we're estimating the location in the time course of these big electrical currents. They're known as primary currents or current dipoles that generate the extracranial signal. These primary currents are generated by the postsynaptic intracellular current flow in the long and spatially aligned cortical pyramidal neurons. Essentially, the alignment and orientation of these neurons within the cortex creates this massive electrical current that produces a magnetic and electrical field that we can record outside of the head. So given that this is where the signal comes from, there are some key canonical features of cortical circuitry that we should be including in our model if we want to study the circuit level generation of this signal. The first is that the cortex is a layered structure. There are synaptically coupled excitatory and inhibitory neurons throughout the cortical layers, including these pyramidal neurons with these long apical dendrites that extend through the layers. In awake behaving brains, these cortical circuits aren't sitting in isolation, but they're constantly receiving input from other parts of the brain. And there are two primary pathways of input to the cortical structure. One pathway is the pathway that relays sensory information from the periphery through the thalamus and up to the granular layers of cortex, where it then propagates to these pyramidal neurons in the other layers. There's another pathway that comes right into the supragranular layers, and this is the information that's relayed from either higher order cortical structures, or there's also what's called non-lemniscothalamic nuclei that project right up to the supragranular layers. So we have our cortical column, and we have these two layered distinct pathways of information flow to this cortical network. Now again, this primary electrical current dipole comes from the intracellular current flow across the whole population of pyramidal neurons. And with this construction in the model, the units of measure that we get out of this simulated current dipole are current over distance, nanoampere meters. This is the exact same unit that comes out of our source localization technique. And so we now have one-to-one comparison between the output of our model and our source localized MEG or EEG data. We know something about the size of the networks that contribute to the most commonly measured MEG and EEG signals. Typically, when you use MEG or EEG, you're looking at either event-related potentials, the evoked response from a sensory stimulation, or low frequency oscillations in the alpha, beta, gamma band. ERPs are typically on the order of 10 to 100 nanoampere meters in size, and they're believed to be generated on the order of approximately tens of thousands of synchronously firing pyramidal neurons. Low frequency rhythms are much bigger, on the order of 100 to 1,000 nanoampere meters, and they're thought to be generated by on the order of 1 million sub-threshold pyramidal neurons. So just to give you a little bit more detail about what's included in our model, we have synaptically coupled multi-compartment pyramidal neuron models and single compartment inhibitory neurons. We calculate the electrical activity in each of the compartments of all the cells using Hodgkin-Huxley current balance equations. So we have things like sodium, potassium, calcium that are regulating the electrical activity of the cells, and then we use cable theory to estimate this intracellular current flow. And again, the net activity out of the model is this intracellular current in units of current over distance. Now, we don't simulate the entire brain. Instead, what we do is we generate trains of action potentials that represent input from other parts of the brain, either the thalamus or higher order cortex. And these action potentials, we can define the pattern of activity of these driving inputs. They activate excitatory synapses in the local network to drive current flow up and down these dendrites. For example, suppose I have a sequence of what I call feed-forward drive. This is going to commit, it's going to activate these synapses, and it's going to drive current flow up the dendrites. That would create a positive deflecting signal in my source localized data. If I had a feedback input that contacts the distal dendrites, that's going to activate synapses out here and drive current flow down the dendrites. Now we're also driving inhibitory neurons, and there's local interactions, and so it's the influence of these exogenous inputs on the current flow within this local network that creates the signal that we're trying to study. Okay, so this is a reduced schematic of the actual network. The full network represents a patch of cortex with a scalable number of pyramidal neurons in the supragranular and infragranular layers. It's in a 3-to-1 ratio with these inhibitory neurons. And our software is specifically designed to study the origin of ERPs in low frequency brain rhythms. Now this is a large scale simulation, right? There's thousands of differential equations, there's tons of parameters. And unless you have experience, all of the code is open source, it's available on GitHub. But if you don't have any experience in building and working with complex neural models, the code is really hard to understand. And so to make this accessible to a broad community, we've embedded it in a graphical user interface. And we've created a website that contains instructions for how to study some of these commonly measured signals, ERPs, and oscillations. We give example data sets building from our own studies and example parameter sets to start to work with. And I'm going to go through how this graphical user interface can be engaged with at the end of the talk. But what I want to show you now is how we've applied this neuromodeling framework in my lab where we've been studying somatosensory perception. And so I'm going to switch to talking about our studies of somatosensory perception and their involvement of these beta frequency oscillations. And so we've been studying perception and performing tasks where we give a brief tap to the finger and we ask the subject, did you feel it or did you not feel it? And what I'm showing you here is data from MEG experiments, but we see similar data in EEG experiments. And our main question of interest has been on studying signals from the primary somatosensory cortex. So we use our inverse methods to isolate the contribution from the hand area of S1, the primary somatosensory cortex. And this is the spontaneous signal from that area when the subject is sitting with their eyes open, waiting for that tactile stimulus. If we put a frequency filter on this signal, we see that it has a strong component in this 15 to 29 hertz beta band. When I tap the finger, we get a desynchronization of that beta activity. And one of the main questions we're interested in is, does this pre-stimulus beta power impact the ability to feel that tap to the finger? So the power is always changing in time. Is the level of the power predictive of whether the subject will feel that brief tap? And we always keep the strength of the tap so they feel it half of the time. And it turns out that this pre-stimulus beta power is a really powerful predictor of detection, such that the higher the power, the less likely the subject is to feel that tap to the finger. What I'm plotting here is I've averaged the pre-stimulus power over the 1 second time window, over the frequency window from 15 to 29 hertz, in sorted power from low to high power. On the y-axis is the detection rate, is the hit rate as a percent change from the mean. And so lower is less likely to detect, higher is more likely to detect. Higher beta power is a signature of inhibited perception. And so the real question is, why? What is it about the mechanisms that create these beta oscillations that cause, when it's there, and a sensory signal comes in, the subject says, I didn't feel that tap to the finger. And so to address this question, we're going to now turn to our computational neuromodeling framework. We're going to use our model to study the mechanisms and how they influence the filtering of this tactile information. But before we do that, we have to take a step back and we have to understand our data a little bit better. And there are some key questions that we have to ask about the data. The first is, what do these beta rhythms look like in unaveraged data? The majority of studies, and all of the ones that I've just shown you, were looking at average responses. We average across time, we average across the frequency band, we average across subjects, but the brain isn't averaging. On any individual trial, we either have some level of beta expression or we don't. And so we want to understand, what is this beta expression in unaveraged data? When we have that information, we can then ask, okay, so what features of this expression are contributing to these functionally relevant differences in average power that we see? Higher average power, I don't feel the tap to the finger. From there, we can try to understand what the circuit mechanisms are that are creating these features in our signal. And then finally, how do these mechanisms translate to, I didn't feel that tap to the finger? And so by answering these questions, the punchline that I'm going to arrive at is that in keeping the output of our computational neural model in close agreement with our data, we've come up with this new theory that the circuit level dynamics creating these beta oscillations, on the circuit level dynamics creating these oscillations, that predicts that the decreases the salience of sensory stimuli through the recruitment of inhibition in the supragranular layers. So we're going to have layer-specific recruitment of inhibition, and that is what decreases the relay of sensory information. So now I'm going to walk through our investigation of each of these questions. What does beta look like in unaveraged data? Well, it turns out in unaveraged data, these beta oscillations are very transient. They typically last less than 150 milliseconds. And so what I'm showing you here is 10 example trials of unaveraged data in the time domain and the frequency domain. And you can see that these beta events, which I've highlighted in red, are very transient in time. Now, because in the spectral domain, these signals are always positive, when you average this, you get an accumulation. And it looks like there's this ongoing continuous oscillation in the average signal. But if we unpack that a little bit, we see that that's not the case on individual trials, and beta events can occur at different times in this pre-stimulus window. This is a very robust phenomena. We've observed it in MEG tactile detection experiments. We've observed it in attention experiments. It's also consistent in the local field potential in mice from the somatosensory cortex. I have a collaboration with Chris Moore here at Brown University, and they've been doing similar tactile detection experiments in mice. And so there's this very robust phenomena that's conserved across species in recording modalities. What I'm showing you here is just some quantification of these transient events. We looked at the number of events in the pre-stimulus period, the power of these events, the duration of these events, and their frequency span. And there's remarkable homology between the human and the animal data, which suggests there's some very robust phenomena creating these oscillatory events. We also have taken a closer look at the waveform shape underlying these beta events. And so here what I'm showing you is we go into the spectral space, we find a high power beta event, and then we go back to the temporal domain, and we're just overlaying these high power beta events. And when we do that, you see that they have a very stereotypical waveform shape that's defined by this negative deflecting peak that lasts 50 milliseconds. And importantly, this is why we have a beta oscillation, high power beta event in our spectral space. The frequency is determined by the duration of this peak. This is not a sinusoidal oscillation with a period of 50 milliseconds. This is an event that lasts 50 milliseconds. And that's why we call these beta events. So we've answered the question, what does beta look like in unaveraged data? They're just transient, and they have this stereotypical waveform defined by this 50 millisecond negative deflecting peak. So now we can ask, well, what features underlie these functionally relevant differences in average beta power that we've seen? Remember, on average, if beta is there, I don't feel the tap to the finger. So now that we know these events are transient, there's several features that could contribute to high average power. It could be that on trials where we have low average power, we have just one event. So this is a schematic of time and frequency. Compared to on trials where we have high average power, we had more events, but of the same amplitude. So if I average this, I'm going to get high power. If I average that, I'm going to get low power. Or it could be that the amplitude of the events is actually different. And so on high power trials, we have higher power events, and on low power trials, lower power events. It could be that the duration of these events is different, or that the frequency span is different. Any one of these differences will create a low versus high power signal in the average. And so we ask the question in our data, which is it that's contributing to differences in power? And it turns out that it's actually the number of beta events that correlates most with average power. What I'm showing you here is the correlation coefficient, Pearson's correlation coefficient, correlating the number, amplitude, duration, or frequency of the events in the pre-stimulus period with the average power. And this is a box and whisker plot over 10 subjects in our human EEG detection task. And here I'm looking at the mean power sorted from high to low and the corresponding event number. And you can see, and this is all of the data from all of our 10 subjects, and you can see very clearly that the highest correlation is with the number of events. And again, this phenomena was conserved across different tasks. So this is our human detection experiment. We've also done attention experiments, and we've also, again, looked at this in mice. So this trend is conserved across species and recording modalities. When we have high power, it's not because we have a higher amplitude oscillation. It's because we have more of these transient events in the pre-stimulus period. So the next question is, well, is that what is controlling the perceptual impact of these events? Maybe that's true for average power, but maybe what really matters for perception is that you had a higher amplitude event or a longer duration event. And it turns out the number of events is what's predictive of perception as well, such that there's a greater number of events in the pre-stimulus period on non-detected or non-attended trials. And so here, we're looking at the number of events, their power, duration, or frequency span as we're looking at the hit rate. This is what I showed you before. This is the probability perception as a function of the number of events, their power, duration, or frequency span. And what you can see as the number of events goes up, the probability of feeling those events goes down. And this was the only significant difference that we saw in our data. When we sort over detected and non-detected trials, we have more events on non-detected trials. And again, same phenomena in our attention task. When you're paying attention, you have less events in the corresponding representation. When you're not paying attention, you have more events. And it was also conserved in our mouse detection task. So a very robust phenomena. What this says is that when you pay attention to your finger, the probability of seeing these beta events in your finger map goes down. And when that probability goes down, you're more likely to feel that tap to the finger. In non-attended representations, the probability of seeing these events goes up. And that increase in beta corresponds to, I'm not going to feel that tap to the finger. Okay, so what features underlie functionally relevant differences in power? The number of beta events is greater during non-detected, non-attended trials. And non-detected this I didn't go over, but the timing of the events also matters. The closer the event occurs to when that tap comes in, the more effective it is at inhibiting perception. So non-detected trials have more recent beta events to that tap to the finger. So now we can turn to our model and ask, what are the circuit mechanisms that are underlying these differences? And why do those mechanisms correlate with perception? And so here we're going to go and use our HumanNeocortical column model, What we're trying to do now in this model is to reproduce the stereotypical waveform that underlies our beta events. Now, this is a very large-scale complex model, and so you have to have a method to constrain the parameter space. And so our method was as follows. The first thing that we did is we built the anatomy and the physiology of the individual neurons. We based this on a lot of literature from animal studies in somatosensory cortex. And then we fix all of the parameters and equations that regulate the individual neurons. We then built in the local connectivity structure of this network with excitatory and inhibitory synapses, and we tune these synapses so we get realistic excitatory and inhibitory ratios of activity in the local network. And then the only parameters that we were adjusting to study the origin of these beta events were the timing and the strength of these exogenous inputs that activate the local cortical circuit, right? So we have these spike trains that activate these proximal and distal dendrites to get activity in this neural network going. And through a long history, years of trying to get this right, we came up with the following hypothesis. These beta events emerge from the integration of a broad proximal input that's simultaneous with a strong distal input, and this distal input lasts 50 milliseconds. Okay, so we have two bursts of activity that come in and they hit the network. If this one comes in and it's stronger and it lasts 50 milliseconds, we'll get a beta event. And so to unpack how that comes about in the model, this is our model simulated beta event coming from the intracellular current flow in these pyramidal neurons that creates this high power signature activity in our frequency spectrum. This is a histogram of the driving spikes, these proximal and distal inputs that activate the local network. If I only had this proximal input, this input would come in and it would drive the current flow up the pyramidal neuron dendrites, and I'd get a single positive deflecting peak like this. But at the same time, this strong distal input comes in, it breaks that current flow, pushes current down the dendrites for one beta period of approximately 50 milliseconds. And with this mechanism, we had very nice agreement between our model data and our MEG data. And we were fortunate through collaboration to be able to test this theory with invasive recordings in both mice and monkeys using these laminar probes where we can get a representation of the activity across the cortical layers. I don't have time to go into that validation in this talk, but this is published work that you can look into more detail in, and I'm happy to answer any questions. Okay, so beta events emerge from this broad proximal excitatory drive that's simultaneous with this strong distal drive that lasts one beta period. Now, I haven't talked about where these drives come from, but there's a lot of support in the literature that suggests that they might come from thalamic nuclei. Thalamic nuclei are positioned to create this simultaneous input to both the proximal and distal locations of these neurons. So now, finally, how do these mechanisms translate to, I don't feel that tap to the finger? When this mechanism is present, the subject doesn't feel the tap. Well, the first thing we did was we looked at our MEG data, and what we see here, we're looking at the evoked response from that tap to the finger, right? So you tap the finger, you get an evoked response in the primary somatosensory cortex. This is what the response looks like when I average over trials where I detected the tap, and when I average over trials where I didn't detect the tap, here in red. I also sorted the data on trials where I had a pre-stimulus beta event and on trials where I didn't have a pre-stimulus beta event. And there's remarkable consistency between whether you had a beta event and when you didn't have a beta event and you detected the trials, or when you did have a beta event and you didn't detect the trials, suggesting a direct causal relationship between a pre-stimulus beta event and the filtering of that tap to the finger. So, how do beta events influence this evoked response? And why does that correspond to, I don't feel the tap to the finger? And so for this, we go back to our neural model. And going back to our neural model, we have to think about, how do I simulate that tap to the finger, right? I've talked about how we simulate this beta event, but how do we simulate an evoked response from the tap to the finger? Well, this is actually one of the first studies that we did with this neural model. And I'm just going to walk you through how we did that. And so the first thing we do is we load in our evoked response data from our human subjects. This is the S1 evoked response. And then we went back to the literature. There's a lot of literature on the origin of sensory evoked responses in primary sensory systems. And that literature suggests that this evoked response is generated by a sequence of activation of this local cortical circuit that consists of a feed-forward input from the thalamus at approximately 25 milliseconds, a feedback input from higher order cortex at approximately 70 milliseconds, and then a re-emergent feed-forward input at approximately 125 milliseconds. So we have input from through these feed-forward and feedback projection pathways that are coming in at certain places in time. And so we could get into our model and test the hypothesis that this sequence of input can generate this S1 ERP. And so at the top of our GUI here, I'm showing you the sequence of activation of the network. Here's a proximal input at 25 milliseconds, a distal input at 70 milliseconds, and another proximal feed-forward input at around 125 milliseconds. When I hit run simulation and these inputs activate this local network, we see that it can reproduce in the model the waveform of this S1 ERP. Now, importantly, the literature guided us as to what this input pattern should be, but we spent a lot of time tuning the strength and timing, exact timing of these inputs in order to reproduce this ERP pattern. Now, and because this is a model, we can also go in and look at the spiking activity of the individual neurons across the individual layers. I'm not going to get into that in this talk, but that information is there, and it's discussed in our early publication. One other really interesting thing is that this same sequence of input, this feed-forward, feedback, feed-forward drive, can recreate evoked responses in other primary sensory areas. So here's a brief evoked response in S1, A1, and V1. And you can see that there's differences in these evoked responses, but they all start by going up, and then you have a large deflection down, and then you go up again. And what HNN is showing, again, by tuning the exact timing and the strength of these drives, is that the same pattern of input can create these evoked responses in these different brain areas, suggesting that at the macro scale, things are really dictated by this common structure of cortical circuits that has these big pyramidal dendrites and these layer-specific pathways of information. Okay, so now we're going to go back to the question at hand. We have beta events. When I have a beta event, I don't feel the tap to the finger, and I get this signature response in my evoked response that looks different than when I do have a beta event or I don't feel the tap to the finger. So how are these beta events influencing the evoked response? And why does that correspond to I didn't feel the tap to the finger? Well, then we went to our model, we simulated a beta event, and we simulated the tap to the finger. We put them together, and what we see is that the model can reproduce these signature differences in the ERP, which is really quite remarkable. And the thing that was even more remarkable is that we actually did this in our model first. I've given this talk like, oh, we looked at the evoked responses in our MEG, and then we built them in the model, but actually we had a beta event prediction in the model. We had a prediction on the origin of the evoked responses. We put them together, and this is what the model predicted should be happening. And then we went back and saw this in our data, which really speaks to when you get the right biophysical detail in the model, it can be generative of new and testable predictions in your data. So what's going on in the model? Why is it that when I simulate a beta event in the tap, I get a response that looks like this? And when I don't, I get a response that looks like this. And how does that correspond to the subject saying, I did or didn't feel the tap to the finger? Well, without going into a lot of the detail, what the model is suggesting is as follows. Remember, these beta events are created by this strong input to the supragranular layers. This input is also recruiting inhibitory neurons in these supragranular layers. When you have a lot of inhibition out here before you tap the finger, then this feedback input that creates this negative deflection in the ERP, it can't get in. And if it can't get in, this signal is going to be stronger. The overall spiking activity in the pyramidal neural populations will be decreased. And if the overall spiking in this primary sensory area is decreased, then the signal never gets out of the primary somatosensory cortex to get to the rest of the brain, and the subject says, I didn't feel the tap to the finger. So again, a beta event creates a lot of inhibition. When there's a lot of inhibition and you tap the finger, the signal never gets to S1. If the signal doesn't get to S1, it doesn't get out of S1, and then the subject says, I didn't feel it. Just to show you that in a little more detail in the model, what I'm showing you here is the spiking activity, histograms of the spiking activity in the different populations of neurons in the model. We have excitatory pyramidal neurons and inhibitory neurons in layer 2, 3, and layer 5. When I don't have a pre-stimulus beta event and I simulate that tap to the finger, I get lots of spiking in the network. And it creates a current dipole signal that looks like this. When I have pre-stimulus beta activity, it recruits inhibition. So that then when that input arrives, you get lots of inhibition and you get no response in the rest of the network. You get a smaller amplitude ERP. The signal never gets to the rest of the brain, and the subject says, I didn't feel a tap to the finger. So how do these mechanisms translate to inhibited perception? Well, beta is decreasing the salience of this sensory input via the recruitment of supragranular inhibition that decreases the relay of sensory information to the rest of the brain. Okay. So that brings me to the last part of my talk, where I want to just briefly give you a tour of our graphical user interface and describe how you use it in practice. I'll touch on some other applications of HNN and some ongoing expansions, and then I'll be happy to answer any questions that you have. So here is our software. When you start the software, you get a schematic representation of the local network and these input pathways from which you drive the network, depending on your simulation experiment. The first thing that you would do is you go in and you can load your data. And here I've loaded the data. This blue trace here is the data from our tap to the finger. Then you set the parameters of your simulation experiment. You do that through the Set Parameters button. And again, we have tutorials that start to teach you about, well, how should I be setting the input to this network to simulate an ERP or a low frequency oscillation? We could define the evoked inputs. And again, here is that evoked input pattern that you would define by pressing this dialog box, which will open the parameter sets to define that input. Then you would hit Run Simulation. This input activates the local network, and then you get the output of the model shown here in gray. Now there's some stochastic nature of this input, and so the gray lines here are the simulation on different trials, and the black is the average. Now, if you want to test a hypothesis, say, okay, here's the ERP from my healthy population, but maybe I'm also studying the tactile evoked response in children that have autism spectrum disorder, and they have some somatosensory differences and some sensitivity to tactile stimulation. And I might hypothesize, well, that's because of the local excitatory and inhibitory balance. And in the somatosensory networks, there's more excitation or more inhibition than excitation. I can go in and I can change the strength of the inhibition and excitation in the local network, rerun my simulation, and see, does the change in the evoked response from that parameter manipulation, is it consistent with the change in the evoked response that I see in my data? So you can go in and you can change various parameters in the network. We can also go in and not only view this current dipole signal, but we can look at the layer-specific responses in the network. We can look at the spiking activity in the different cells in the network. We can look at the frequency responses of this network. And you have lots of different things that you can visualize in the software. Some other applications of HNN that we're working on and other groups have worked on, we've been looking at the impact of non-invasive brain stimulation on neural circuits by looking at the impact on the EEG signal, and then using HNN to study, well, what's creating that difference in the EEG signal before we give the non-invasive brain stimulation and after. And here was a study where we gave 10 hertz alpha frequency alternating current stimulation. Some groups have been looking at differences in circuit mechanisms underlying increased beta activity and decreased gamma activity in children with autism spectrum disorder. Some groups are looking at changes in EEG signals with depression and the effect of ketamine for depression. Other groups are looking at changes in EEG signals that are happening with development. And so all of these different applications HNN can be applied to all of these applications to study the circuit mechanisms underlying differences in either EEG or MEG signals that emerge. Some of the ongoing directions, we're currently integrating this neural modeling software with the source localization software, specifically M&E Python, that's being spearheaded by my collaborators, Matti Hamalainen and Alex Gramfort. And so the idea is that you'll have one software package where you can both localize where the signal came from and interpret the circuit mechanisms of the signal. We're integrating it with other neural modeling platforms so that we can expand this template model to have different cells and different circuits and expand the utility of the software overall. We're also looking at ways to simulate and visualize things like LFP, CSD, ECOG. All of this are ongoing work in the lab. And so that brings me to my conclusion. I've showed you that HNN's foundation is a neocortical column model under thalamocortical and cortical drive. It simulates the primary currents underlying EEG and MEG signals based on their biophysical origin. We've embedded this complicated model in a user-friendly graphical user interface, and we have tutorials for how to study ERPs and low-frequency brain rhythms. HNN combined with MEG and inverse recordings in animals revealed this novel mechanism on the origin of these transient beta events that's suggesting that beta is decreasing the salience of sensory input through the recruitment of supragranular inhibition. And the HNN software can be accessed through this new website that I encourage you to take a look at. And hopefully I've convinced you that this software can be applied to uncover circuit mechanisms underlying EEG and MEG correlates of healthy and neuropathological states and aid in the development of new treatment strategies, either with pharmacology or with non-invasive brain stimulation. And so with that, I want to thank you for your time. I also want to thank my lab and my collaborators. My research goals require collaboration, and I've been really fortunate to develop a network of interactions that allow me to pursue this interdisciplinary research. I have a phenomenal lab. Everybody has contributed in a meaningful way to the research that I presented. The HNN development team is a multi-institutional effort between researchers at Brown University, the Martino Center at MGH. My collaborator, Matti Hamalainen, is creating the M&E inverse solution package, he and Alex Gramfort, and then at Yale University, the Neurons software development team. And so with that, I will end, and I'm happy to take some time to answer any questions, or review any slides or pull some more up. And I know this is a lot of information in what feels like a short period of time, but thank you again for the opportunity, and I'll now be happy to take any questions. I'm not
Thank
sure
you
how.
very much.
Okay. Can you hear me?
Yes, yep, no
Yes,
problem.
okay. While we are waiting for connection from Alex. Alex, are you
Yes,
ready?
hi, I'm here, Stephanie, I'm sorry. Yes,
Hi guys.
No worries.
Yeah,
Alex, are you en route home? I understand that it's like 5 p.m. there, so.
no. I'm in the car now. Actually, I'm on
Okay.
sort of a vacation. I'm trying to make it. But anyway, let's go back to your talk. Okay. We have the reason I delayed because I was switching from the questions panel to where I can turn on my mic. We have several questions already, and let me try and ask them to you. So that the first question is rather technical. It asks about the hardware that is needed to run the simulator. And another associated question is it Mac only? So that's the question.
No, okay. It does run on any platform, and the installation instructions are on our website. It runs on Mac, it runs on Windows. Linux is the best place to run it,
Sure.
obviously.
I agree.
But yeah, there are instructions. It's easiest, again, on Linux, but for Windows and Mac, we've been suggesting that people run it through Docker, a Docker hub, but all of the instructions are there. It's also available, and we've created it, it's so that hopefully it runs on everybody's laptop or desktop computer. We've had good luck with all the people in our group being able to run it on their machine, but it's also available through Amazon Web Services if you wanted to run some really, but you have to pay for that.
Yeah. And you have to, for the web server, do you have to sign up for it, or what, or how does it work?
It's freely downloadable. It's distributed through GitHub, and yep, anybody can download it. There are some licensing agreements. It's free for academic use, but not for commercial use.
I see. Okay. Then the next question is, alpha is often associated with the information inhibition. I believe information perception inhibition. Did you observe any relationship differences between alpha and beta in your studies?
Yeah, we have observed some interesting differences. So I didn't focus on alpha in this study, but in S1, we see this alpha beta complex. And the model is predicting that this complex is, again, coming from these two inputs. And on any cycle, there's a stochastic nature of these inputs. And if they come in together, you get a beta oscillation. If they come in slightly off, you get an alpha oscillation. And so we actually simulate this with two 10 hertz inputs to the local network. And when they come in together, you get beta. When they come in a little bit off in any phase, you get alpha. What we see in our data is that when we give an intentional cue, pay attention to the finger, ignore something distracting in your foot, alpha rhythms are engaged immediately. But when you have to make the decision about whether you're going to feel that tap or not, beta rhythms seem to be more important. And so we have a paper from 2010 and also 2015, where we look at the temporal relationship of these alpha and beta changes in this cued attention task. And we see that there are some differences in the timing on when these alpha rhythms versus beta rhythms come into the experiment to shift attention or to actually tell me whether you felt that tap to the finger or not.
I see.
So there are some interesting differences. We haven't looked a lot at the waveform shape of alpha to try to verify if our model prediction about these two inputs is different. There's some support in the literature that it is, but for the past few years, we've been focusing on beta.
Okay, thank you very much. Yeah, I think this is a very, very exhaustive answer. And well, another question, how sensitive is HNN to a number of EEG, MEG sensors? Would it result with reasonable results from 4 to 8 sensors already, or is in beamforming 64 plus sensors are needed?
So that's a really good question. And it really depends on the experimental paradigm. But one thing I want to make clear that when you first hear about HNN, it's hard to kind of get your mind around. What HNN is actually simulating is not the sensor data. We assume that you've already done your source localization. And what you're giving to the HNN software is not the EEG sensor array or the MEG sensor array information. You're giving to the software these source localized current dipole signals. And so we've separated the problem of the inverse solution, has its own issues. And you have to trust that your inverse is accurate. And then once you have that assumption, now you use HNN to study that circuit level mechanisms, creating whatever signal that you're putting in that from your source localization. Now I say that, but we've actually used HNN to study sensor level EEG signals without source localization. And in this non-invasive brain stimulation study, we were just looking at the sensor level EEG signal from the tap to the finger. It turns out that the way this evoked response looks at the EEG sensor level and the way that it looks at the source level is nearly identical, which makes sense because it's one underlying circuit that's creating the current dipole and the sensor level signal. And so we do think that if you only have sensor level activity, HNN is still useful. The tricky part there is relating positive and negative directionality to whether you're going up or down the dendrites. But the signals do look the same. Beta events that we measure on EEG sensors look just like beta events that we measure in source localized signal. But with the source space, we have directionality. We know which ones are going out of the cortex and which directions going into the cortex. At the sensor level, you have to infer that information.
I see. Okay. Okay. Thank you. Okay. Then another question: do this model corresponds to spontaneous beta events or only for beta events after stimulus?
Everything that I've talked about today is spontaneous pre-stimulus beta events before we tap the finger. Now, sometimes it's after an attentional cue, right? So I say, pay attention to your finger, but we're still considering those spontaneous pre-stimulus beta events before we actually have them report detection of a stimulation.
I see. Okay. Another question, but I think you spoke about it, but maybe I missed. But okay, is it possible to model ERD in HNN?
Sure, but you have to think about what ERD means. ERD means the beta events go away. And so you want to think about what's the actual question in the ERD that you want to address. And then what's the right simulation experiment to address that question? HNN is really designed to match to waveform shapes, right? And so if you saw a difference in the waveform shape after you have this, when you have this desynchronization, then that's something that you could start to understand where that difference in shape comes from with HNN. The whole power is the matching one-to-one between the signal and the output of HNN, and we do this on non-averaged data. So ERD is an average measure, right? On average, beta power goes down. First step would be to unpack, well, what does that mean in a single trial? And once I have an understanding of what that means, then I can start to think about how to use HNN to interpret the circuit mechanisms. So probably what's happening is when beta goes away, other phenomena come about, right? When they get the beta desynchronization, there's something else going on in the local network. And so you would use HNN to understand, well, what is going on in the local network? Beta went away.
Yeah. Okay. Thank you. And then there are many questions, therefore I kind of, how many neurons in HNN model?
Oh, that's a very good question. It's something I forgot to touch on. Let me go back to a slide. So I mentioned that these, this is a slide, that these big electrical currents are created by thousands or hundreds of thousands of synchronously active pyramidal neurons. In our model, we don't simulate 100,000 neurons. What we do is we typically simulate roughly 100 pyramidal neurons in the supergranular and infragranular layers. We assume this synchronous activity across a larger network, and we use a scaling factor. We multiply the signal by some factor that tells us, that gives us an estimate of how many neurons are actually participating. And so if I was to look at the output from the 100 pyramidal neurons or 200 pyramidal neurons, 100 in each supragranular and infragranular layer, the signal would actually be really small in comparison to my recorded data. But I take that signal and I multiply by a scaling factor. Here I've used a scaling factor of 3,000. That will give me an amplitude that's consistent with my recorded data and an estimate that on the order of 3,000 times 200, 60,000 cells are actually contributing to this signal that I recorded. So that's a really good question. You can change the number of neurons in the model, but at the end of the day, it becomes intractable and maybe not necessary because everything's doing it together, or else we wouldn't even see the signal.
I see. Okay, thank you. Yeah, there is another thank you, but don't go away. It's just a thank you from Tommy Krutkovsky, who asked the question about the number of channels. He thanks you for wonderful answers and wishes you a good night from Tokyo. Okay, so it's, you see, it's the Pan World seminar, so everybody, the whole world is here. Okay, then another question.
Really
from
unbelievable when you think about it.
Yeah, it's amazing. Yeah, it's amazing. And I'm sitting in my car somewhere, I don't know, in the south part of Russia, you know. So that's pretty cool. Okay, can we another question, can we simulate ripple like up to 100 hertz, or there is activity, or there is some physical limit in the spectrum allowed by the software?
No, there's no physical limit. You can set so when we do, so the model is simulated on like 0.025 millisecond time scale. So I guess that would set the resolution of the frequencies that you would be able to numerically simulate. But when you want to do the frequency analysis, there's a panel where you set the frequency range over which you want to calculate frequencies. The default, I think, is from 0 to 100 hertz, but you can change it from 0 to 1,000 hertz. It uses a Morley wavelet to calculate the frequency spectrum, but there's no reason why you couldn't study high frequency signals.
Right. I see. Okay, then there are no more questions in the chat, but I have a couple of questions to you as well. So did I understand correctly that one of the, well, the first question is that the HNN, it doesn't have the sort of optimization tool, right, which would try to feed the parameters to the data, right? So it's up to the experimenter to tune the parameters, right, and then observe the similarity between the measured waveform and the one that is generated by the HNN.
Another very good question that I forgot to touch on. We do have some parameter optimization, but right now it's only for ERPs. And so the way that it works is you first get a good representation of your signal by defining the number of evoked inputs that you want. You start with our tutorials. You get some kind of rough idea from playing with the inputs to the model where the ERP would come from. Then you can define parameters that you want to optimize over once you have a good understanding of these are the parameters that really matter. You're not going to optimize over all the parameters because there's too many of them. But you can define a subset of parameters, and there's an optimization tool. I wonder if I have it in this GUI here. We also have a methods paper on this now that describes how to use the optimization tool. In the tutorial on our website. Oh, sorry, I went too far. No, we don't have it here, but there is under these menu buttons, there's an optimization button where you can optimize. But again, right now it's only to evoked responses.
I see. Because the impression I got. Yeah, sorry, go ahead.
We're working to make it so that it goes on any signal. Ultimately, I'm realizing what the community wants is they want to put in a waveform and they want the software to tell them how is this created in the neuron.
Yes.
Now, I originally thought that's an impossible, that doesn't make any sense, but now I'm rethinking things because there really are only so many signals you get out of the head, right? You get ERPs, you get low frequency oscillations. they tend to have canonical shapes, which I'm now realizing through studying more and more signals. If that's true, then we may be able to target specific waveforms, specific oscillations, and give people this tool that they want, which is put in a waveform that has a shape that we kind of know at least the prior space of where it could come from. And then we can optimize on that prior space in the model to tell you how the brain generated it. So we might get there, but right now it's more of a hypothesis and development tool with some limited optimization.
I see. Okay, great. And another question: did you try to look into intellectual spikes, into spikes which happen between the seizure onsets, right? Between the seizure events, and did you try to model them? Because or at least HNN can be potentially applied for that, because it would be very interesting to maybe it will allow to somehow disentangle the seizure onset, the epilepsy mechanisms, or to understand what's wrong with the brain tissue that is generating these spikes.
I think that's a really exciting and fascinating and perfect question for HNN. We have not looked at it. There's a couple of groups that we've started, trying to collaborate with to look at that, but it hasn't gone very far. What I found in practice, this is a new software. It was used in my lab for 20 years, but we just in the past 3 years, with some funding from the National Institutes of Health in the US, have had the money to turn it into this software with a graphical user interface that people can work with. And so we're just starting to work with other groups. But what I find is anything takes a targeted person for this to be their project, right? And
of course.
If you want, we're going to study, have a collaboration to study intellectual spikes or to do this or that, there really needs a person to be dedicated to it who's going to learn the model, apply the model, work with the data, and we just haven't had that collaboration for this yet.
We may actually try to develop this kind of collaboration, because we've been looking quite closely into the spikes, but we were primarily looking into, well, first I was of course looking into detection and all this kind of technical issues, but then we started looking at trying to represent the spikes as traveling waves, and we have some very interesting observations that are not yet published but are out there on bioRxiv, and so probably we can then chat later, maybe when you have time, or via email, so we can exchange some ideas, and maybe we can come up with a collaborative idea. Okay, I have another question actually from my friend and colleague Anton Chizhov, who is doing brilliant work on simulation of neuronal populations with simple models, but to tackle the activity of the entire population. He's experimenting now a lot with slices, and so with brain slices. And so his question is, did you try to validate the model with data from slices? Do the software has options to visualize signals recorded by page clamp, for instance? And also, do you also multiply synaptic currents on a scale factor similar to what you do with the number of neurons?
Well, I'm sorry, what was that second question? Do we what?
The second question, do you also multiply synaptic currents on a scale factor with a scale factor similar to what you do with the number of neurons, similar to what you use with the number of neurons? Yeah.
Okay, so starting with that first question about are there ways to visualize things that you could validate with slice recordings? We're actually in the process of writing a grant right now with Barry Connors Brown, who does slice work, and describing exactly how we would do that. You can get somatic voltages out of the different pyramidal neurons. That's an option in the software. I think I show it right here. Use somatic voltages.
Interesting.
It's there. You could also look at spiking activity of the cells. We've built now, it's not actually distributed with the software yet, but we have it, a way to look at local field potentials and current source density across the cortical layers. And so there are some ways to validate this with slice data. The question about scaling the synapses, the answer is no. You can scale the synapses through this EI button, this synaptic gains button that I'm showing here. If you want to test predictions about changes in the synapses, you can also go in and you can, in this local network button, you can change any of the synaptic strengths that you want in the network. The network was tuned to get realistic behavior out of this cortical column. So we tune the EI balance, we tune the spiking properties of the cells so that we didn't get, epileptic activity when we start to get input into the circuit. But what you get when you load HNN is our template model. It's been tuned, it's been validated in several ways, and then you go from there. So if you want to change the synaptic gain along the populations, you can absolutely do that through this button. So I guess the answer to the question is yes. This button right here is the scaling factor for the synapses. But you can also go in and tune individual synapses through this local network button that will bring up all the synaptic connections.
Okay, and then yet another question from Anton. Do you agree that in rough approximation, the extracellular potential is just proportional to the difference of membrane voltages in somatic and dendritic compartments?
I'm sorry, can you repeat that question?
Yeah, sure. Do you agree that in rough approximation, the extracellular potential is just proportional to the difference between the membrane voltages? Okay, I will read it as it's written. Is just proportional to the difference of membrane voltages in somatic and dendritic compartments?
I don't think it's proportional to the membrane voltage. I think it's proportional to the current flow in and out of the membrane at those locations, right? Because we got current flow. Inside the cell, current will go in one direction. Outside the cell, current will go in the other direction. So the extracellular potential is going to reflect the difference in the current flow at those two different locations. Not necessarily the voltage, which may have which may not go in the same direction as the current flow. It could in many circumstances, but I don't think it has to in every circumstance.
I see.
That's my intuition.
Yeah. Okay, thank you. And then another question, basically it also relates to Anton's work, because when we were looking at these integral spikes and we were looking at trying to describe them as waves, we found that they propagate at the speed of approximately 1 millimeter per 1 millisecond. Okay, so basically 1 meter per second. And when Anton was simulating his data, because he had several papers on simulating the integral activity and pre-equal spikes as well, so he found that the speed of propagation was about 10 times slower, and it's consistent with his measurement that he's making with slices, okay, with surviving slices, okay? So my question is, do you think that the difference that we observe between what we see in integral spikes non-invasively and with what Anton has seen and simulating with his with slices, could it be that there are some connections, some extra, or either connection to some different brain regions or to some subcortical structures that mediate this propagation that we see non-invasively? And the continuation of the question, whether or not this can be modeled with HNN.
Yeah, so to answer the first question, I would say absolutely. I mean, I really don't know where that slower time constant comes from, but when you cut those connections, we know those connections are critical for the current flow that we get in awake behaving brains. And so that would be a natural place where that difference might come from. Can you right now in HNN, you cannot look at wave propagation. There were some things that we'd have to do to the connectivity structure within the network in order to look at that. And we're, like I mentioned in one of my last slides, we're trying really hard right now to make the model so that we can use models developed in NetPine. NetPine is a simulation platform that's been designed to easily to build network models.
Okay.
And if we can create HNN so that you can use NetPine models, then we could address the question you have more readily, because we could create a model that has the right connectivity structure to look at wave patterns of activity. But right now, that's not possible in the model that's distributed with HNN. But again, all of the code is on GitHub. And we're trying to, the other thing we're trying to do is be software developers, which is not in my background of expertise. I'm learning a lot. But trying to do this so that we get a community of people involved in expanding and developing the software. And so if there are any software neuromodelers out there, please join us in our efforts to make this software more amenable to questions like propagation of current flow.
I see. Okay. Yeah. Thank you very much. Yeah, I think we exhausted the first layer of questions. Okay, and we need to sleep over it and think more. And I will be very happy if you consider a possibility of answering questions which will probably arise in the nearest future, and so that we can perhaps establish some sort of collaborative interaction with your team and you. So what the
Absolutely.
thank you very much once again from me and from the crowd who was listening to you. There were I'm trying to see, there was at some point there was 53 participants, so I think this is a large number, at least for this 4-hour seminar. We tried to advertise it. So, and we once again want to thank you for finding the time to present and for the excellent work that you are doing. Yes, and actually I would like to also mention that Stephanie has recently received a prestigious BioMag award, which I think is a very, very reasonable award that to justify the brilliance of work she's doing. Yeah. So yeah.
Well, thank you all again for this opportunity. It was really exciting to be able to speak to you all in other parts of the world, and we're eager to collaborate with people on getting HNN in useful applications, and so please do be in touch. I'll be happy to answer further questions or email, and we can be in touch, Alex, to talk more about more specific collaboration.
Okay, great. All right. Thank you very much. Yeah.
All right. Thanks, everyone.
Thanks.
Stay well.
Thanks, Stephanie. Thank you. We'll try. Yes. Okay.